Welcome to This Explainer. Today, we're diving into something really incredible. We're looking at a process that literally drives both modern biopharmaceuticals and our green energy future, all based on a fantastic foundational presentation by Chem on ion exchange chromatography. We're talking about the incredibly high stakes of separating completely invisible charged molecules out of chaotic, messy liquid mixtures. So you might be wondering, how do we actually separate the invisible? I mean, this is the ultimate challenge, right? How do you isolate specific, microscopic ions, say a life-saving protein or maybe a crucial trace metal, when they're just swimming around in this complex, messy soup of impurities? Well, you need a mechanism that operates way down on the molecular level, relying entirely on the fundamental laws of physics and chemistry. And the answer to that? It's ion exchange chromatography, or IEX for short. Simply put, IEX separates ions and polar molecules based on their reversible Coulombic affinity to an oppositely charged ion exchanger. It sounds super technical, but think of it kind of like microscopic Velcro. Opposite charges attract and stick together. So if you have a positively charged target, you're going to use a negatively charged stationary phase and vice versa. It's a highly targeted, localized charge interaction. Here is our quick roadmap for today. 1. The magnetic attraction principle. 2. Meet the resin matrix. 3. Setting the stage with buffers. 4. The art of elution. And finally, 5. From proteins to green tech. Okay, let's dive right into this with Section 1, the magnetic attraction principle. The whole core of this process relies on a direct inverse relationship. In cation exchange chromatography, your target molecule is a positively charged cation. To catch it, we've got to use a stationary phase that is negatively charged. On the flip side, in anion exchange, our target is a negatively charged anion, which requires a positively charged stationary phase. Basically, the target molecules are drawn to the stationary phase like a magnet, while all the similarly charged or neutral impurities just flow straight through the column unhindered. Moving along to Section 2, meet the resin matrix, the chemical heroes of our story. Now, what's really interesting here is how we categorize these resins based on their functional groups. The resin is essentially a column packed with tiny, insoluble beads that have either weak or strong exchange capacities. But here is the crucial part. Strong and weak actually don't refer to how tightly they bind. It's about how well they maintain their charge across different pH levels. A strong ion exchanger, using something like a sulfopropyl functional group, stays fully charged over a massive pH range, roughly from 2 all the way up to 14. A weak exchanger, like DAIO cellulose, only maintains its charge within a much narrower window, usually between pH 5 and 9. All right, Section 3, setting the stage with buffers. Let's look at the actual operational flow of running an IEX column. We can actually condense a pretty dense lab manual into a fast, dynamic 4-step sequence. Step 1 is equilibrate. We run a low-salt buffer through the column to establish our baseline pH. The real trick here is that the buffer has to put our target protein at least 1 pH unit away from its isoelectric point. That's the exact point where the protein has a neutral net charge. And if it's neutral, it's not going to stick. Step 2, load the sample. This is where those oppositely charged target molecules bind to the resin. Step 3 is wash, where we just keep filling the buffer to wash away all those non-binding impurities. And finally, Step 4, elute. We change the conditions to force the target molecule to let go so we can finally collect it. Which brings us to Section 4, the art of elution. I absolutely love this quote from a biochemist describing the process. They said, "I'm exchanging the salt and then pushing off the protein with the salt. It's going to kind of like compete." You see, elution is essentially the ultimate chemical game of musical chairs. Since our target molecule is bound by an ionic interaction, we can break that bond by bringing in a competitor. Usually that means we slowly increase the salt concentration in our buffer. All those salt ions flood into the column and start competing with the target proteins for the charged spots on the resin. As the salt level rises, it eventually outcompetes our protein, literally bumping it off the resin, so it washes out the bottom of the column for us to collect, perfectly purified. Now let's zoom out a bit for Section 5, from proteins to green tech. So why lithium? Well, continental brines currently supply a staggering 59% of the overall global lithium. And with the completely exploding demand for electric vehicles, our current production methods just aren't fast or efficient enough for the future. We absolutely need better recovery processing. Scaling up this microscopic ion exchange process into massive, industrial, direct lithium extraction plants is literally critical for the green energy revolution. To optimize this, Researchers from the Department of Energy evaluated different ion exchange resins specifically for pulling lithium out of complex brines. They compared two proprietary resins, resin A and resin B. The data showed something fascinating. Resin A only successfully extracted lithium when the brine's pH was adjusted to exactly 7, but resin B, it could extract a lithium at a pH of 6. Now, why does a single point on the pH scale matter on a huge industrial level? Well, to adjust a massive natural brine pool up to a pH of 7, you have to pour in vast amounts of caustic chemicals. Because resin B works at a lower pH, it drastically lowers that caustic consumption, making it far more economically and environmentally viable at a massive scale. And this brilliantly illustrates the data. Looking at the Pareto chart analyzing extraction performance, you'll see two massive bars towering over that red significance line, time and temperature. In the incredibly complex, messy chemistry of lithium brines, this mathematical model proves that time and temperature give us our vital signal, while almost all the other variables are just statistical noise. By running these statistical models on the ion exchange process, researchers summarized the real-world application with an exact recipe for success. To get the absolute maximum lithium recovery from this specific setup, you need to run the extraction for exactly 8 hours at exactly 55 degrees Celsius with a 0.2 gram resin dose. By mastering these microscopic, highly specific factors, time, temperature, and pH, we are literally dictating the economic viability of the global battery supply chain. It's a real game changer. Which leaves us with a truly fascinating thought. We've seen how opposite charges allow us to pull highly specific, invisible molecules out of chaotic mixtures. We use it to isolate proteins for life-saving biopharmaceuticals. And we use it to pull lithium right out of the earth to power our cars. So, if we can master the invisible attraction of ions at this microscopic level, what else can we extract from the noise? Thank you so much for joining me on this explainer, and keep questioning what's hidden right in front of you.